tell you things that you need to know, so. Usually you get this written down. Format will be, as it was the last time, won't be any different. Courses will be, or the questions will be along the same line, so. This one, 3 chapters, will probably have a few more questions on it. I'll try to keep it within 50, 55, but it may have questions, but it may have 4 points.
We're not going to know anything from Unit 1, like that?
No, just the ones 4, 5, and 6. So, I'll tell you the main things, and when we have a class, that's only 50 minutes. I have to be limited on the number of questions that I can give, which I don't really like, but that's the way it is. You have to have enough time to be able to get all of these done. So, you have to be done at, well, waiting. I have classes and things right after this, and so I haven't had time to get to it. So, it has to be done. First one here, that we started to look at was carbon. There's gonna be some things about all of these that you need to take out with you, that you really need to remember. And, we will try to tell you all those. Carbon is the backbone of organic molecules. One of the reasons that it's important is in organic chemistry, the definition of this is the study of compounds that contain carbon. Just remember, not all compounds that contain carbon are going to be looked at necessarily in organic chemistry. Notice the 4 things that organic compounds will contain. Atoms. It says, we looked at those: carbon, oxygen, hydrogen, and nitrogen. It says most organic compounds contain hydrogen atoms in addition to carbon. They also contain the oxygen, and they have to have they don't have to have oxygen and nitrogen, but usually in some form or in a functional group, whatever they will. Remember, the key thing to what makes carbon so valuable as an organic compound, it has 4 bonding sites. This gives it a great amount of versatility in what kind of molecules it can then make and what can bond to it. Yeah.
She said you can't hear you.
Oh. Let me move towards the front of the room. I said the big thing about carbon or bonding sites gives it great versatility to be able to make other compounds and to bond with other molecules. 4 valence electrons. They can make double bonds or even triple bonds. Everything that we've talked about, carbohydrates have carbon, lipids, carbon, proteins, carbon, so they can make a lot of variety when it comes to different molecules. One of the things about it is the shape that it has. If you notice, like with methane, when these bonds form, they're not just evenly spaced out. These form at different angles, and the particular shape that you see right there, like where the methane is a tetrahedral and so forth. Because of where its electrons are spaced, where they're located, it has covalent bonding ability and Notice these four that we had mentioned before. Carbon is with hydrogen, oxygen, and nitrogen for most molecules. And it is because of that bonding where their electrons are, how many shells they have, so forth, that makes them so easily to bond. And as we've said, carbon can form double bonds. That's the bonding arrangement of carbon dioxide, for example. Yeah.
You said that it can form single, double, or even triple bonds, right?
Correct. Most, there will be two forms to carbon compound. They can either be in a chain or in a ring. These are example of the chains, these are example of the rings. And if you take something that we looked at such as glucose, remember glucose, it's in powdered form, it's pretty much in a chain, but when it does go into water and dissolve, it goes into the ring form. Also remember benzene. Just remember that it is called an aromatic carbon compound. And there are many things that will bond to it and make many different kinds of chemicals. You don't have to remember the structure or the configuration, just that benzene is a ring and that it's aromatic. We talked about hydrocarbons. Those are the ones that have, obviously, the hydrogen and the carbon is attached to the carbons where there is a bonding site. They contain in those bonds a lot of energy. Whenever those bonds are broken, they release a lot of energy. That's why fats contain so much energy. Fats are stored in adipose cells. These are the fat droplets. Here's the nucleus of that cell. Now notice this part all around it is the cell. These are just droplets of fats contained within the cell. If we take, and we will see this in the next chapter, but if we take a chain of the hydrogens and carbons, hydrogens attached to the carbon, then that's called fatty acid. It's got a carbonyl group on it. But a straight chain like this is just called fatty acid. Right here. When we put three of them together with a glycerol molecule, then it's called a triglyceride. We mentioned the isomers. There's not that much I want you to remember totally about isomers, except notice they are compounds that have the same molecular formula, but their arrangement is different. And because it has that different arrangement, they can have different properties. These are the three kinds: structural, different covalent arrangements of the atoms; cis trans, same covalent bonds, but in different places; enantiomers, isomers that are mirror images of each other. You should know what the three are, yeah.
So we should just know what's on the slide about those. Thank you.
The two that you really need to know are the cis trans and the enantiomers. Remember, the enantiomers is just a mirror image. It's easy to remember. The cis trans, they are in different places compared to this double bond. If you use the double bond as your center point, then you can see here the things that are added are different. They have different arrangements, locations as compared to that bond. What you do need to remember more than anything is the enantiomers, and remember these versions, L and D versions, biological molecules, one version may be active and the other one is not. That's the big thing to remember. And this tells you a little bit about them. Yeah.
You said in biological compounds, yes, one's active and one may not be. Okay, thank you.
That's it. Yep. Here it gives you these versions. Don't try to remember anything on that. We're talking about chemical groups and how they can affect things. The number one is the functional groups. They are called functional groups because they do have different functions. Whenever you put them on a molecule, they will act in different ways. You need to know all of those. And you need to be able to give the configuration of these whenever, or the way they're written, like the OH carbon group, C double bonded oxygen. Carboxyl group is a carbon group with a hydroxide attached, so forth. Now, this is the one that we had talked about with the Zwitter ions. Notice it says acts as an acid, can donate hydrogen because the covalent bond between the oxygen and the hydrogen is polar. The big thing to remember is the non-ionized form is the way it's written right there. When it is ionized, and that hydrogen proton leaves, then we get this form. And that's when it becomes the Zwitter ion. Many of these functional groups you will see written differently, and make sure, even like the amino right here, notice the non-ionized form is NH2, but if it's ionized in that a hydrogen proton is added, it becomes then this configuration, NH3. So you may see it written either as NH2 or NH3, depending upon whether they are talking about its ionized or non-ionized form.
So for the test, we need to know both ionized and non-ionized?
Yep.
Would we write that as, like, in?
You
with
can write it as
positive charge?
NH2 or NH3, or you can write it in that configuration there.
Okay, should we put the positive charge by the end if we just write it out? Instead of just NH3?
You should, because these functional groups have charges. Most of them are all negative. Now, if they become in the ionized form like this, then, you know, that can change. Another one to look at, phosphate. Phosphate can be written as just PO4, or it can be written as OPO3. Once again, based upon its form in through there. The thing I want you to remember about this is the fact that these protons, ions can form, protons can move from place to place and so forth. So their ionization can change. And you need to look at this right here and understand a little bit about it. This is the one thing. This is a methyl group. Now remember, methane itself is CH4, but we have to remove a hydrogen, have a place for that to bond to something else. So now it's CH3. It's lost a hydrogen. It's going to, or an electron, but it's going to make up for that hydrogen by bonding to something else and share an electron then, and it will get back to being stable again. But whenever you add a methyl group, CH3, to another molecule, it is said to be methylated. Methylation is important. Notice here, primarily. addition of a methyl group to DNA or the molecules bound to DNA affects the expression of genes. That is methylation of DNA. It can change the expression of the genes. Arrangement of methyl groups in male and female sex hormones affects their shape and their function. So remember that term, you'll see methylation pop up a lot. Notice it is just simply a group. And when you can add that group then to other things, and it will change the function of it. And that's the same way with all of these. If you go back and you look at hydroxides make alcohols, the carbonyl groups. You notice right here make ketones, carboxylic acids, that you need to know. What do those functional groups make when they are added to a molecule? Alcohols, ketones, carboxylic acids, amines, sulfhydryls, thiols. Phosphate is organic phosphates and methyl is methylated compounds. So you need to understand those. We said adenosine triphosphate. That's a phosphate molecule that's attached to an adenosine, which has two phosphates already attached, so now it becomes ATP-free. ATP is the organic molecule, adenosine, 3 phosphate groups. You need to know that, and as I said the other day, you need to be able to draw that, just a little box. You can draw the phosphates, remember, as a P inside of a circle. Was good enough to draw the phosphates. There it is. You could just leave this stuff out, just put the P's there. You can leave the oxygen. And notice another thing about most of these functional groups, they have a lot of oxygens involved. Whenever you see something like phosphate, it's got oxygens. Sulfate, sulfur with oxygens. If you take an oxygen away from a sulfate, then it becomes sulfite. So when you hear terms like phosphate, sulfate, it's referring to the number of oxygens that's attached to that. This shows the ATP molecule. Whenever it loses the phosphate, it's all finished. ATP is the energy carrier. Remember, think of ADP, adenosine diphosphate, as a battery that doesn't have a charge. And when you charge it, it becomes ATP. Then it can give off energy. So that's the things to remember about carbon. Make sure to remember and know those functional groups fairly well.
Maybe a question about the process. Does it become like the inorganic phosphate because it's losing the carbon when it reacts to H2O?
When you look at this, you're getting back to the ionization. Now, what were you asking right here? What were you asking?
I was just asking about the inorganic phosphate. Does that happen because it's reacting and losing a carbon when it like reacts with the H2O?
Well, basically, what's attached to. Guys, the question here was like the form of the or the phosphate separated from carbon. There's many of these elements. Take nitrogen, for example. Now, nitrogen makes up about 70% of the atmosphere that we breathe. Plants have to have nitrogen in order to grow. When you look at a plant and you see green, that's coming from the nitrogen a great deal. The problem is a plant itself cannot use nitrogen in the air. Nitrogen in the air, remember, is a diatomic molecule, N2. So plants can't use that. The form that they can use it in is a nitrate. That's when the oxygens are added to it. Put the 3 oxygens around NO3. Then they can use that form of nitrogen. Plants cannot convert into it can, I should say into. It cannot convert diatomic nitrogen into a nitrate. Plants can. It relies on bacteria to do that. Those bacteria are in the ground and they are called nitrogen fixing bacteria, which means they put it together with the nitrate. That way plants can use the nitrogen. Some plants, like beans, are called legumes. They can take the nitrogen straight out of the air and use it. They don't have to have nitrogen fixing bacteria. So when you look at these things and you hear terms such as nitrates and sulfates, phosphates, all of that, they are referring basically to the oxygen that's attached to that element. And it makes it a more usable form for most organisms. Now, if we just have a PO4 molecule, the electron arrangements are different. It will become that OPO3 when it's attached through there. So it does change the arrangement, not the actual molecule, but the arrangement. Yeah.
Do we need to know this like formula?
Yep. I mean, you should know it already. ATP, you lose the phosphate, you get ADP and energy released. So that's basically what you should know. Chapter 5 was the big one. Instead of going through every single little slide, I want to get a few things here for you. Remember, you need to know what a polymer and a monomer is. And you need to know and be able to actually draw a dehydration reaction. And remember the little violet balls up there represent a monomer. That monomer could be a simple sugar monosaccharide if it's making a carbohydrate. It could be amino acids if it's making a protein. They could be nucleotides if they're making DNA. And what would they be if they were making a lipid? They won't, because lipids, remember, are not polymers. We said out of the 4 molecules that we looked at in this unit, carbohydrates, proteins, lipids, and nucleic acids. Carbohydrates, proteins, and nucleic acids are polymers. Lipids are not. We gave another name to a chain of carbon that's got hydrogen attached to it. What was that?
Are we talking about hydrocarbons?
Hydrocarbons, yeah. Remember, all a hydrocarbon is just a bunch of carbons. Well, carbon's not a monomer. It's not a monomer, it's just an element. When you attach the hydrogens to it, then what it is called is a hydrocarbon. You should also remember, in a dehydration reaction, dehydration means lose a water molecule. What's that? When we become dehydrated, you lose water. Dehydration reactions make a polymer grow, it adds a monomer. And it has to remove a water molecule so that these ends up here will be exposed, so the other monomer can then come attached to it. So to expose this end, what has to happen? To expose that, this has to lose the hydroxide on that end, this has to lose the hydrogen, that makes a water molecule. Now the ends are exposed and the bonding can take place and the polymer can grow. Also remember, a polymer will have a hydrogen on one end, the hydroxide on the other. Guys, this is just as simple as learning Russian. Don't outthink yourself. I'm telling you what's going to be on the test. These little things. It's important to remember a polymer has a hydrogen on one end and oxygen on the other, because so do these monomers. Notice that monomer has a hydrogen and an oxygen. Yeah, Brooke.
Is it hydrogen on one end and oxygen on the other, or
Hydroxide. Hydroxide, a functional group that we looked at. So hydrogen on one side, hydroxide on the other. They then, whenever they're removed, can form water.
But monomers are also the same. Every
Monomers-
single one will have a hydroxide in it.
Whether it's a polymer or a monomer, there will be a hydrogen on one end, a hydroxide on the other. They are the same, like that. And remember, removing a water molecule is the way you attach a monomer. I promise you, I'll ask on the test, what has to be removed to make a polymer grow? The water molecule. Now, the opposite takes place if we are going to break apart. If we've got a big monomer, like a big complex carbohydrate, polycarb, in order to break these apart, we have to do the opposite. Now we've got the polymer right here. So if we are going to take this monomer off, let's say that is glucose from a carbohydrate. We're breaking it apart so your body can use it for energy. When that is broken apart, look what has to happen. We've now messed up the octet rule and neutrality of the electrons and the charges. So we have to have water come into the system, add a hydroxide, and then add a hydrogen here so those two can separate.
So does hydrolysis take place when H2O is like the solvent? Is that when this would happen?
Does it take place when?
When like H2O is the solvent, like that's when this takes place, hydrolysis?
It's in a liquid environment, yeah, but, that's not the reason it's taking place. It's because we have to satisfy the electron configuration on each of these, the monomers and the polymer. But does it take place in water? Yeah, we wouldn't have, energy cellular
That
respiration.
was kind of the reason why I was asking whether like the cell could do this, like only if there's H2O already present, or if it has H2O and it knows it's because of this, so it's raining through water.
There is, it makes one, remember? Well, you don't remember, because we're not making it. But what Terrence was asking here, where does water come from, basically? Cells, as we will find, whenever you burn energy, they make water. Look. Right there. When we tear apart those carbohydrates, or if we start to make them, we've got water. Well, as we go through at the end of this, water comes into the system, then we break it apart. This is going to go through cellular respiration, this individual monitor. It's gonna be broken down, we start to pull electrons, protons, things away from that molecule in order to make energy. At the end, we return both the oxygen and the hydrogen back. We've used it, and it comes back together and makes water again. So it's kind of regenerating.
Oh, that's what I was just gonna ask. So it's basically like a cycle. When it breaks down polymers, it makes water. It extracts the water from the bonds basically, and then it uses the water later to break polymers for energy.
It doesn't use the water to break it, but it has to have the components of water to be on each end to satisfy the electron configuration. And then, as that monomer, like glucose, goes through your system, it's gonna start to be broken down. At the end, we are gonna be left with the way that this energy, the force that is going to be used to make ATP is coming from the hydrogen protons, not the electrons. You're gonna have hydrogen protons build up on one side of the membrane, and then it's gonna be pulled back through the membrane, and that creates a force that will make ATP. So then we've got this hydrogen sitting in the middle of the cell with nowhere to go to. When you breathe oxygen in, then you put oxygen back in your system, it combines with the hydrogen to make water, and it can move out of the cell. That's why you have to have oxygen. Really, it's the only reason you really have to have it, as far as breathing, is just to be able to collect those hydrogens at the end and make water. Macromolecules, remember the big ones that we talked about, the carbs, the lipids, the proteins, nucleic acids, these are big molecules. We've talked about polymers, remember sugar is the building block, individual sugars, monosaccharides are the building blocks of carbohydrates, amino acids are the building blocks of proteins, nucleotides are the building blocks of DNA. This talks about monosaccharides. Remember, the most basic sugar that a plant makes will be in the form of CH2O. So then it can start putting those together. If we put 6 of them together, then we've got C6, 6 times 2, H12, 0, oxygen, 6. C6, H2O6, which is the formula of glucose. So all, basically what I'm saying, if we multiply every bit of this by 6, then we get glucose. So if we put 6 of those together. Yeah.
So basically, you were saying that amino acids make up proteins, nucleotides make up DNA, monosaccharides, the simplest form is CH2O for that.
Monosaccharides make up carbohydrates. The simplest kind of sugar is the CH2O.
Thank you.
And we said on sugars, there will be a carbonyl group, that is the carbon double bonded to oxygen. Where it is placed on that carbon skeleton will determine, is that sugar called an aldose? If it's on the end, it's an aldose. If it's on the second carbon, it is a ketose. You do not have to try to remember these. One thing that you should basically remember are these simple monosaccharides. Not all of them, but you should recognize glucose, lactose, fructose, ribose as monosaccharides. And if you start to put these together, you can get things like table sugar, sucrose. I think the lactose and glucose makes lactose. You don't have to remember all that, but just when you put these together, then you start getting disaccharides like table sugar and milk. And remember, they form rings in water. I want you to be able to draw glucose. And folks, it's real easy to remember. think of the formula, 6 carbons, right? These are kind of your rules for drawing glucose without going nuts. 6 carbons, hydrogen on one end, a carbonyl group attached on the other end. So you've got hydrogens basically here on both ends, but you've got a carbonyl group on the other. Then, all you have to remember, 1, 2, 3, 4. On the 4th carbon, we will have a hydroxide to the right, so forth. There is a change in through here. So, the easy way to remember, 6 carbons, hydrogens on both ends, carbonyl group on one end, then starting on the 3rd carbon, you got all hydrogens on one side except for the 3rd carbon, which has a hydroxide, and it's just a reverse on the other. And look at fructose. It's a little bit different because it's ketose, carbonyl groups down here. But it still has almost the same arrangement as far as the hydrogen and hydroxide. Yeah,
I can't go back here, but, the orange square, is that a double bond?
the orange bond, yes, that's double bond. I can blow this up a little bit, make the people in the front go blind. You'll see a lot about sugars because that's the basic fuel. Remember, you just have to do them linear. You don't have to draw the ring shape. You're going to need, the things you'll need to draw, basically, you're gonna have to be able to draw adenine or adenosine, ATP. You just draw the box which says adenosine, 3 phosphates attached to it. You will have to draw sugar, glucose, and you will have to draw a nucleotide when we get to that. And all the nucleotide is adenine plus the sugar plus the phosphate. You can still use the little square boxes. You don't have to draw the chemical arrangements of those. Do remember that these ring structures represent, these points represent carbons. The type of bonds, when we bond 2 sugars together, it's called a glycosidic linkage. When we bond 2 proteins together, it's called a peptide bond. When we bond fats, the fatty acid change to a glycerol molecule, it is called an ester, E-S-T-E-R bond. I'm gonna have to move through here quick because we'll run out of time. You don't really need to remember these structures. Like glucose and glucose makes maltose, glucose and fructose makes table sugar, but you don't have to remember that. Polysaccharides are long, complex structures, a lot of monosaccharides and a lot of linkages. Remember, starch is an animal storage carbohydrate. In animals, glycogen is the storage polysaccharide. And also remember, the primary place we will find glycogen in mammals is going, in other vertebrates, will be in liver and muscle cells, because that's the ones that's going to use the energy the most and provide. Liver will provide, break down that glycogen and prepare it to go through the system, and muscle cells will use it as well. Remember, cellulose is unique because it has 2 kinds of bonds, alphas and betas. We can break the one, but we can't do the other. Can't break the beta. shows the bonds on cellulose. Enzymes to digest starch by hydrolyzing the alpha can't do the betas, so we can't digest cellulose. Some animals will use microbes, such as cattle, ruminants, things that can eat the woody stem plants and survive. And they do it because they have in their system bacteria that can do it. Chitin, remember, is another form of polysaccharide, and that makes up the exoskeleton of both arthropods, insects, and so forth, crabs, whatever, lobsters, and also the cell wall of fungi. That shows the locust shedding its skin. And we said chitin is also useful because it is carbohydrate from this stuff. It makes up the skeleton of these things. They can also be used to make surgical thread, which then your body will reabsorb and make it go away, break it down. Lipids, know the basic structure of the lipids, hydrogen and carbon. Fats are lipids that are solid at room temperature. Oil are fats or lipids. They are lipids that are liquid at room temperature. So a fat is solid. A fatty acid contains a carboxyl group attached to that carbon skeleton right here. That is a fatty acid chain. There's the carboxyl group. Now, when it attaches to a glycerol molecule, once again, we're attaching something there, so we have a dehydration reaction. When we attach 3 of these, then that bond that forms there is that ester bond or ester linkage. When we put 3 of those fatty acids on the glycerol molecule, we have a triglyceride. And that's the same as the triglycerol. So remember, on a carbon fatty acid chain, if everywhere there's a carbon and everywhere there is a hydrogen that could be attached, as long as we have all of the hydrogen attached and fills up all of those carbon chains that we can, it is called a saturated fat. If, on the other hand, we have a hydrogen missing, the carbon will then have to form a double bond in order to satisfy its electron arrangement, and then we get this bond or dip in the molecule. That then makes it unsaturated, which means it takes up more volume, more space than what a single one would. Because of that, it's got less energy. Yeah.
Do we need to remember the structure of that?
No. Fats made from, you need to know there's a double bond. So,
Thank you.
fats made from saturated fatty acid, as we say, solid at room temperature. Plant fats, fish fats are usually unsaturated. Now, I'd say most fats, animal fats and things, are saturated and solid at room temperature. Fish fats, plant fats are different, and they should be really saying lipids more than fats there. Notice it says another name for oil is an unsaturated fat, so they are all, however, lipids. If we add hydrogen to a unsaturated fat, that is called a trans fat. The process is called hydrogenation, where you just basically pump that liquid fat and add extra hydrogen to it. Trans fats do not break down very easily at all, particularly in your body. Some unsaturated fatty acids are not made in the body, some are. The ones that we have to have in our diet will be things such as omega-3, which we need for growth and immune system and so forth. And because our body does not make it, we must get that from food. And there are certain things, particularly fish oil, will have a lot of omega-3, which is one of the reasons they recommend eating fish basically once a week to make sure you get enough omega-3. It can be in some plants, but olive oil is good and so forth. But we do require it because if we require it in the diet, it is called essential. That means it's essential that it is found in the diet, in our food. Phospholipids, remember, are one of the biggest uses of lipids in our body. You need to be able to draw that little membrane that it makes where you've got just two of these basically end to end. It's got a phosphate on one end, then it's got two fatty acids tables. That makes this membrane. So if I tell you to draw it on the test, you can basically just draw this right here in the fatty acid tails. Phosphate, you should be able to label this as the phosphate end, and these as the fatty acid tails. Steroids are fats, lipids, I should say, not fat. Cholesterol is a kind of steroid. Cholesterol is important, we need it in our bodies. And when we start to study this membrane, it will start to show little pockets of cholesterol in there. And the purpose of that is when the weather gets cold outside and the temperature drops, it prevents your cell membranes from just getting hard and packed and dense. It keeps fluidity in that membrane so it can still function. That's why your body makes cholesterol. Proteins, there are many different kinds. You should know this right here. They include support, structural, storage, transport, communicating, movement, defense. Those are some examples of the proteins, what they do. And notice it says they're more than 50% of the mass, dry mass of most cells. Remember, proteins end in IN. Enzymes end in ASE. And we also mentioned the terms catabolic. That means to break down a big molecule into smaller ones.
What was that again?
Cat, C-A-T-A-B-O-L-I-C, catabolic. Breaks big molecules down to small ones. Anabolic does the opposite. Takes small molecules and then makes bigger ones. Enzymes, remember, are protein catalysts. They make other reactions occur at a faster rate. Polypeptides are polymers built from, we only have 20 amino acids. 10 of those our body can make. We've got genes that will code for it. There are 10 that we cannot make. That must be in our diet. Those 10 that are in our diet are called essential amino acids. A polypeptide is a long polymer. Whenever that polymer becomes functional, it's a polymer of amino acids. When it becomes functional, then it is called protein. You should be able to draw that right there. That is a generic amino acid. It's got an amine group attached to a carbon, attached to a carboxylate group. A hydrogen, and then it will have some group attached here that will make the amino acids have their own individual characteristics. There are 20 different ones. The only thing that's different is the R group. That is going to be the functional group. So you should be able to draw that. That first carbon is called the alpha carbon. You do not have to remember the proteins. Remember, they can be mixed as far as polarity. And some can be either polar or non-polar. You don't have to remember them. Note at the end of a polypeptide, there will be one end that is a carboxyl end, and then the end terminus. Notice carboxylic end where the carboxylic acid is, and then the end terminus, the amino group.
So it's carboxylate, one's acid.
Carboxylic acid will be on one end, that's called the C terminus. The end that has the amino group is called the N terminus. Remember these structures that we talked about, there's four structures of protein. Primary, where it's just a chain of amino acids. Secondary, where it starts to get contorted, twisted. Tertiary, they become folded and make bonds. Remember the types of bonds that can be made, that can have a covalent bond, and then it can have weaker bonds such as hydrogen bonds, van der Waals forces, and so forth. A quaternary is just two or more tertiaries put together. And this shows the structures of these. You can read up on those. Most of those covalent bonds that are going to be formed will be these disulfide bridges right there. This shows hemoglobin, and it just shows each one of these subunits could be a tertiary protein that comes together to make the hemoglobin. Remember, denaturization is changing the shape of a protein. The function of a protein will be determined by its shape. If we change that shape, we have changed its function. These proteins are coated, they have active sites where other things can attach. When you straighten them out, denature them, that site's no longer there. Chaperonins are just little protein structures that help a protein to fold correctly. DNA, we talked about deoxyribonucleic acids and so forth. Remember, messenger RNA is a copy of the gene. Transfer RNA will take amino acids to the ribosome. The ribosome is called rRNA. So the ribosome itself is a type of RNA. There's the ribosomes, and then out in the cytoplasm, amino acids here will be brought in by transfer RNA. Read up on this. We've run out of time, but it's pretty straightforward. Know what a nucleotide is, and this is the basic one I said you should be able to draw. Just, you're going to have a base, an adenine, for example, just say nitrogen base, a sugar, and a phosphate. And you can draw those just little square boxes. You don't have to try to draw those shapes. Understand, it is the phosphate and the sugar that makes the long strands of DNA. The bonds that form between the nucleotides, those type of bonds will be hydrogen bonds. So read through this. A nucleotide is when we have the nucleoside, the nitrogen base and the sugar, when we add the phosphate group to it. And on the cells, just go over what we had talked about, and you should be in pretty good shape. The cells are going to be general. Know what the organelles are, what they do, and we'll be good.
Did you sign this?
Nope.